Good evening to all. I'm presenting before you some very informative kind of report which we have done, that is diagnostic, prognostic and predictive protein-based biomarkers, which we are basically doing for early detection of cancers, that is liver and lung cancer, and as well as neurodegenerative diseases, which includes Alzheimer's, Parkinson's and epilepsies. So as I have asked to be fast, I'll just briefly talk about proteins. Proteins basically are the tools for the discovery of biomarkers, and they are divided into three distinct classes, that is profiling proteomics, functional and structural proteomics. Why we are studying proteins? Because there are 30,000 genes and more than 200,000 proteins. So that means there are one gene which is basically expressing more than one protein, and after the expression these proteins are undergoing post-translational modifications, and that is the reason we have to go understand the protein in a pathological condition and then go through the reverse genetics and identify the genes. And biomarkers are basically the objectively evaluated biological processes. It can be for the pathological interventions, for pharmacological responses to any of the drugs which are being administered to the patients. And they can be also classified into three, that is diagnostic, prognostic and predictive. We are basically concentrating on the early diagnostic biomarkers as well as the prognostic biomarkers in the tumors and brain tissues. This is the basic strategy for a biomarker discovery, where we are taking a pathological condition versus normal and going through the mRNA level analysis for the candidate gene or protein level analysis for the candidate gene, which can be used for diagnostic, prognostic, therapeutic and biological purposes for clinical applications. Basically, the discovery part is interlinked with the validation, which is very, very important for the application of the biomarkers for the diagnosis. Basically, I'll rush through the tools of proteomics, that is the 1D analysis, where we are separating the proteins based on the molecular weight, followed by a 2D, because very many proteins have the similar molecular weight. They cannot be really separated out in 1D, so we go through the isoelectric focusing and separate it in the 2D, where the proteins are totally separated on the basis of isoelectric point as well as the molecular weight. The second strategy is for peptide mass fingerprinting, where we get the peptides fragmented and the masses elucidated. The third is mass spectroscopy 2D gels, where we are also doing the post-translational modification studies to know whether the proteins have undergone any type of post-translational modifications to decipher the conditions. And then it is the 3-dye multiplexing, where the proteins are differentially labeled with different fluoropores, and then they are excited at the different wavelengths to understand what are the proteins which are getting. And this is the basic slide which exhibits the differential expressions, and based on the molecular weight and the isoelectric point, we can differentially know which are the proteins which are present in the samples. Coming to the basic conditions which we are working, that is, one is hepatocellular carcinoma. This is one of the leading causes of cancer-related deaths, and this is the report which is published in 2008, and basically the incidences of liver cancers are increasing day by day due to a lot of pollutions and drug abuse, alcohol abuse, viral hepatitis and so on and so forth. The progression of the disease is initiated by a chronic liver disease followed by cirrhosis and HCC. And once this particular condition is attained, like from cirrhosis still there are chances, but after HCC chances of recovery becomes very, very feeble. So again the need of an early diagnostic biomarker becomes an urgency. The risk factors already I have told you, and the prospective biomarkers till date which are there are again not the proper validated markers. They are basically there for the routine purposes, but they are also in other pathological conditions. So we have to have a specific biomarker for the diagnosis purposes. Keeping these things in mind, we have designed a model for development of liver cancer. That is, this model is a modified Salt-Faber protocol which we have followed, and animal which has been used is the male Vista rat, and a single dose of diethyl nitrosamine followed by the selection phase of 2-acetylaminofluorine CCl4 and again 2-acetylaminofluorine has given us a model within 60 days. Now this is something which is not there. Salt-Faber's model is a 9-month model, and the need for the model was like from the day after the challenge, 24 hours we start the screening of the proteins to understand the difference, and this is basically just a baseline study for exactly understanding whether the model is progressing well or not. This is the slide where it clearly shows that the liver progression to the neoplastic lesions as well, followed by the binucleate cell is there. That means the HCC has started. Coming to the gel electrophoresis, we are doing the differential expressions, and you can see the basic up and down regulations of the proteins very clearly. And this is followed by the 2D where you can directly see in the control versus disease condition what a different pattern, differential pattern we are getting. So we identify these proteins which are differentially expressed in the disease condition and then go for their peptide mass sequencing and followed by the rest of the studies for validation. PDQuest analysis is done to understand which are the proteins which are, how many proteins are up or down regulated. This is the PDQuest study. And the spots exactly identified are extracted out, and then they are taken for the MALDI-TOF mass spectrometry to understand which are the proteins which have been eluted out. And this is the peptide sequence, and then they are followed by the proper sequence and understanding, and this is one of the protein which has been discovered in the case of HCC. It's a liver regeneration protein, and now the studies are for validation in the human subjects, whether the protein is present in the which condition it is there, and so on. This is another report where we are working with the lung cancers, and similarly there are some specific biomarkers in the lung cancers which are already available, but again the validity and the specificities are lacking in these biomarkers. The models we are developing are by the derivative of nicotine NNK as well as nicotine itself, and these both models are worked simultaneously. Basically we are working for the diagnosis by the serum, and the prognostic markers are in the tissues which we are getting in the different stages. Similarly, the baseline studies for the enzymes to just monitor the changes in both, and here also you can see the successful development. The lesions, neoplastic lesions are already there, followed by the differential analysis in the proteins, and a beautiful slide which exhibits up and down regulations of many proteins in both NNK and nicotine based models. Now all these proteins are under the process of validations, and we have already discovered one of the prospective biomarker which we have now got the primers design, and that gene is cloned for it. The gene also has been decided, which I'm not really exhibiting out here. This is the protein which we have sequenced. It is an apolipoprotein. It is a specific protein usually in cases of lung cancer it is upregulated. So basically the thing is, when we are working in the area of medical practice in the 21st century, there is always a need for the prediction, detection and pinpointing for the personalized medicine. So this kind of strategies where we are into the discoveries of early biomarkers, early diagnostic kits before the interventions of any pathology can be a boon to the society in future. Thank you.